Welcome to the deep dive. We take a whole stack of sources, research papers, notes, you name it, and we pull out the key insights for you. Often we find some pretty surprising stuff. Today, we're diving into something people have dreamed about for, well, centuries, directly connecting our brains to devices. Just think about it. You've got these fantastical old ideas like the Nuremberg funnel literally pouring knowledge into someone's head, and then, the much darker stuff like the telescreens in Orwell's 1984, reading and writing thoughts. Wild ideas. But the core problem has always been, well, obvious, right? Our tech is mostly mechanical, electrical. The brain. It's alive, it's wet, it's dynamic. How do you even begin to bridge that gap?
Well, it turns out a really big piece of that puzzle, the sort of missing link, might be nanotechnology. We're talking about engineering things down at the nanoscale, single nanometers up to maybe a few hundred. And that scale, you see, perfectly matches the brain's own building blocks, biomolecules, parts of cells. It's the right size. So where older tech was just too clumsy, too big, nanotech offers this almost bespoke way to interface. It's a whole new ballgame.
Right, that's the key. So for this deep dive, that's our mission, unpacking how nanotechnology is making these, sci-fi dreams feel a lot more real. We'll look at medical breakthroughs, amazing research tools, and yeah, we have to talk about the ethical side too, the big questions that come up when you start talking about plugging into the brain. And what's really striking is just how much progress is happening, often kind of quietly, you know, behind the scenes. Okay, so let's get into it. How do you make an artificial device talk to something as, well, complicated as a neuron? For ages, just making contact without messing things up was a huge challenge. But now, nanotech seems to be changing the actual wires and sensors.
Yeah, the fundamental issue was always size mismatch. Like you said, traditional electrodes,
Yeah.
way too big. Imagine trying to listen to one person whispering in a stadium with a giant megaphone. You get terrible signal quality, you can't get close enough, and you often cause damage, scarring. The brain fights back. Even the gold standard technique, patch clamping, which measures single cells, it's super fiddly, low throughput, and you can only really do it for a short time before the cell is damaged. So nanotech's solution is basically to shrink the interface right down. Think nanostructured electrodes, tiny, tiny structures made of gold, silicon, carbon nanotubes, graphene. These dramatically increase the surface area at that tiny scale, which means lower electrical resistance, much better signal quality, incredible specificity. You can get right up close, even gently pierce the cell membrane to listen inside, or deliver a tiny puff of a drug right where it's needed. And the key thing, these devices are often ultra small, flexible. They cause much less damage, which means they potentially work better for longer.
Okay, better connections, less damage. But getting inside the cell without wrecking it, how does that work? And how do you stay there? That sounds tricky.
Oh, it was tricky. For years, getting a stable, long-term recording from inside a neuron was, well, almost impossible. The holy grail, really.
Yeah.
Sometimes it happens spontaneously, but not reliably. So researchers developed clever tricks to sort of nudge the membrane open. You can use a little electrical zap. Electroporation creates temporary pores, but they seal up fast, limiting your recording time. A really exciting method now is optoperation. You use a very precise laser pulse focused right on the nanoelectrode tip. It creates tiny temporary pores, just enough to get a great signal, and these recordings can last over an hour without harming the cell. It's quite elegant.
Wow, an hour inside a cell recording.
Yeah.
That's impressive.
And building on that, you have things like nanostraws. Think tiny vertical tubes on an electrode array. These can actually extend into the cell, creating this incredibly tight connection, this coupling. And crucially, without needing external force pushing them in. This means super stable, long-term recordings. You can even pick up really faint signals, these things called postsynaptic potentials, which tell you a lot about how neurons are actually communicating.
Okay, so that's getting close without force. Amazing. But what about getting deeper into living brain tissue for, actual in vivo applications?
Right. For in vivo in living animals, nanowires are a big focus. Their tiny diameter is key. They cause much less tissue damage when implanted, so you get less of that immune response, less scarring compared to bigger implants. People have used them to record signals deep inside rat brains, for instance. But it's not just about recording. Their potential is frankly astounding. They could potentially restore lost senses. Imagine a nanowire converting light or even tiny physical strains directly into electrical signals the brain understands, like creating artificial sight or hearing.
Yeah, that's mind-bending. Using light or strain instead of just electricity. The brain speaks electrically and chemically, but you're saying we could use other languages, light, heat, magnetism.
Exactly. But then the big question is, how do you translate? How do you get the brain to understand these other inputs? Take light. Optogenetics is huge, right? Using light-sensitive proteins to turn neurons on or off. But the visible light you often need doesn't go deep into the brain very well, and it can be harmful. This is where something called up-converting nanoparticles, or UCMPs, come in. They're pretty clever. They absorb harmless near-infrared light, NIR, which does penetrate deep. Then they convert it locally into the visible light needed to activate those optogenetic switches, all without needing fiber optics snaked deep into the brain. People have used this in mice to trigger dopamine release, create specific brainwave patterns, even recall memories. It's powerful stuff.
Wow, using invisible light to control brain cells deep inside. Okay, what about heat? Or even pushing on them?
Yep, heat too. You can use heat-sensitive channels in neurons. Nanoparticles, like little gold nanorods, can act as tiny antennas. Shine a light on them, they heat up locally, photothermal stimulation. Just enough heat can trigger calcium signals or even make the neuron fire by changing its membrane properties. Or you can use magnetic fields. Magnetic nanoparticles convert an alternating magnetic field into localized heat magnetothermal stimulation, again activating specific neurons. The catch with heat is that it's generally a bit slower than direct electrical zaps.
Still pretty precise, though.
Yeah.
And mechanical force, you can actually push on neurons.
Sort of, yes. With magnetic nanoparticles, again, sometimes called M-torquers,
Yep.
you apply an external magnetic field, and these particles physically twist or pull on specific proteins in the neuron membrane mechanosensitive channels. We're talking piconewton forces, unbelievably tiny. But what's amazing here is it's wireless, it's fast millisecond timescale, and very localized. They've actually used this to control movement in mice, to manipulate specific circuits deep in the brain just by applying a magnetic field.
Okay, so that's writing into the brain, stimulating it with light, heat, magnets. Incredible toolbox. But what about reading it? Listening in? How do we use nanotools to hear that complex brain chatter more clearly?
Right. Accurate readout is just as vital. You need to
Uh-huh.
listen well to understand what's going on. One way is using quantum dots, QDs. These are tiny semiconductor crystals that act like voltage sensors. Their light emission changes based on the neuron's electrical state, its membrane potential, the big plus. They're way more stable and brighter than older dyes, so you can potentially image activity in real time for longer. The challenge, though, is getting them exactly where you want them and oriented correctly. They're really small.
Okay, so QDs for electrical signals. What about the chemical conversations, the neurotransmitters?
Hugely important. And nanotech has answers there too. For example, tiny sensors based on field effect transistors, FETs. They're coated with specific DNA strands aptamers that bind only to certain neurotransmitters, like serotonin. And because they're so small, they overcome the sort of natural electrical muffling effect in tissue, the W length, technically. This lets them detect incredibly low concentrations, femtomolar levels of serotonin in the brains of live moving mice. It's astonishing sensitivity. You can even imagine this tech leading to, say, wearable stress sensor someday.
The femtomolar.
Yeah,
That's vanishingly small. Are there other ways to listen chemically?
definitely. Another approach uses functionalized carbon nanotubes, single-walled carbon nanotubes, or SWCNTs. These glow in the near-infrared spectrum, which is great because NIR light passes through tissue well with less background noise. Scientists have made a sort of dopamine nanosensor paint with these. You can literally map dopamine release across brain tissue slices with high resolution in space and time. And similar sensors exist for other key chemicals like acetylcholine, often by detecting the tiny pH shift when the chemical breaks down. It's giving us a much finer view of the brain's chemical landscape.
Okay, this tech is amazing, clearly moving fast. Yeah. But it raises a practical point. How do scientists test all these new nanoparticles and interfaces without jumping straight into living animals every time? That seems crucial for, well, speeding things up, but also for ethics.
Absolutely. And that's where these advanced test platforms are becoming so important. Nanotech helps make these in vitro models much more realistic. One method uses organotypic slice cultures. You take thin slices of brain tissue, maybe from rodents or even human tissue from surgery, and remarkably, they keep their structure, their cellular connections alive in a dish for weeks. You get great optical and electrical access. It's a really good bridge between simple cell cultures and a whole living brain.
Human brain slices.
Yeah. And then you have the really cutting-edge stuff, 3D brain organoids. These are often called mini brains. You grow them from stem cells, often derived from a person's skin cells, called iPSCs. These little blobs can develop various brain cell types, complex structures, even something resembling the blood-brain barrier. This is huge. You can test how drugs or nanoparticles get into brain-like tissue, study diseases like Alzheimer's or brain tumors in a human-relevant model, and the exciting part, you could potentially test treatments on organoids grown from a specific patient's cells. Personalized medicine, but for the brain, in a dish. And yes, it significantly reduces the need for animal studies.
Mini brains from skin cells.
Okay, that's incredible. Are there other lab models helping us figure out how brain circuits work? Yes. There's also the brain on a chip, or BOCI approach. These are microfluidic devices, tiny platforms with channels and chambers. You can design them to mimic different brain regions, cortex, hippocampus, whatever, and guide neurons to grow networks between these regions. This lets you study how different parts of a circuit communicate, how patterns change in health versus disease models like schizophrenia. It gives you a controlled way to probe network dynamics.
So you're recreating brain circuits on a chip, and I heard 3D printing is involved now, too, to make them even more brain-like.
Exactly right. The brain isn't flat, is it? Its power comes from its incredible three-dimensional complexity and connectivity. So researchers are using 3D nanoprinting techniques to create intricate scaffolds, little structures within these chips. These scaffolds physically guide the neurons to grow in more realistic 3D arrangements. Getting that 3D architecture right in vitro is vital if we want to truly understand how these networks compute and process information. It's all about the connections.
Right. Okay, so we have these amazing tools, these better models. Let's tie it back to the big goal. Brain-machine interfaces, BMIs. From sci-fi to now, what's the reality? How close are we to really plugging in?
Well, it's important to split BMIs into two main types,
Yeah.
non-invasive and invasive. Non-invasive, you've probably heard of EEG caps measuring electrical activity from the scalp, or fNIRs using light. They mostly read brain activity. They detect the combined signals of millions of neurons firing together, so the information rate isn't super high. Then there's fMRI, functional magnetic resonance imaging. That's kind of the gold standard for non-invasive imaging of where activity is happening. Great spatial resolution, down to millimeters. But it measures blood flow changes, which are slow compared to neuron firing. So you see where things light up, but with a delay of a few seconds. You lose that precise timing.
Okay, so non-invasive gives you the big picture, but maybe not the fine detail or speed. What about invasive?
Invasive means putting the sensors closer to the neurons right on the brain surface, subdural, or actually into the cortex, intracortical. This gives you a much better signal, higher signal to noise, much faster information transfer. And this is where we see some truly mind-blowing real-world applications starting to emerge, like that brain bridge implant you mentioned. It essentially created a digital bypass for a spinal cord injury, allowing a paralyzed person to regain some walking ability by decoding brain signals related to movement and tension.
That's life-changing stuff.
Yeah.
But what are the roadblocks for these invasive implants? I mean, putting things in the brain long-term sounds challenging. Wires sticking out.
Yeah, exactly. Huge challenges remain. The brain's immune system sees implants as foreign objects. It tries to wall them off with scar tissue fibrotic encapsulation. This can degrade the signal over time. So implants need to be incredibly biocompatible. They need to be flexible to match the brain's softness, to minimize irritation. Size matters too smaller, generally means less reaction. And yes, the whole issue of wires through the skull, it's a major infection risk and practical limitation. True wireless operation power and data transmission is a huge engineering goal. Getting these nanostructured electrodes to work reliably and safely long-term inside the brain, that's still a major frontier.
Okay, so materials, immune response, wireless tech. Lots to solve.
And there's another really interesting angle emerging. We talked about the brain speaking chemically, right? With neurotransmitters. So maybe our interfaces should too. This is leading to the idea of chemical BMIs. Devices that don't just sense electrical signals, but can actually detect and release specific neurotransmitters, directly matching the brain's chemical language. There is a device called NeuroString, for example, that can monitor key chemicals in real time in freely moving mice. People are even building artificial neurons that can receive a signal, say indicating low dopamine, and then release dopamine in response, closing a chemical communication loop. It's a very new, very multidisciplinary field, but it holds immense promise for neurorehabilitation, maybe even cognitive enhancement, by tapping into that chemical information layer.
Chemical communication.
Yeah.
Wow, okay, these advances are just staggering. But inevitably, they bring up some really big questions, don't they? How far do we go? What are the rules? It's not just about can we do it, but should we?
You're absolutely right. Neuroethics isn't just an afterthought. It's crucial, and it has to evolve alongside the technology. The potential medical benefits are obviously a powerful driver, restoring sight, movement, communication. It's hard to argue against that. But we absolutely have to think about the ethical boundaries. It's like the old analogy of fire. It can warm your house or burn it down. Brain interfacing tech needs strict ethical guidelines. And these conversations need to happen now while the tech is developing, not after it's already widespread.
So how do we approach that? How do we build in ethical thinking?
Well, there are a few complementary ways. One is ethics by implication, trying to look ahead, forecast the potential social consequences, good and bad, and steer development towards the good. Another is ethics by discovery. This means looking at the inherent properties of the tech as it emerges. For example, if one type of nano interface causes less inflammation than another, that's an ethically relevant discovery, safer is better. This approach often resonates well with engineers.
Building ethics into the design itself makes sense. What about the practical side, the rules?
That's where good scientific practice comes in. Transparency is key. Open dialogue with everyone involved, patients, ethicists, the public, policymakers. We need rigorous testing, long-term safety studies, especially with nanotech, where you have unique concerns. What happens to these tiny particles long-term? Can they cause chronic issues? Can we even get them out if needed? Often, no. So clear regulations are vital. Interdisciplinary work, ethical training for scientists, it's all part of the package. And we absolutely have to address fairness, privacy, data security, and the real risk that these powerful technologies could widen the gap between rich and poor if only certain people can access them.
Yeah, the access question is huge.
And there's one more piece here that's critical. AI and machine learning. These brain interfaces generate enormous amounts of data. AI will be essential to make sense of it. But think about future bidirectional interfaces, reading and writing. You could have algorithms interpreting brain signals and then sending control signals back to the human. Who controls the algorithm? What happens if it makes a mistake? This really highlights the urgent need for legal frameworks before this technology fully matures. We need safeguards against misuse and unforeseen consequences.
Right. Algorithms potentially influencing our thoughts or actions. That's a serious consideration.
Yeah.
So wrapping this up, what's the takeaway for us trying to grasp all this? Well, today we've really seen how nanotechnology is radically changing the game for interacting with the brain. Tiny electrodes that get incredibly close to neurons, using light to switch brain cells on or off, recalling memories. Even this future idea of chemical communication, it feels like the future of neurotech is arriving fast.
It really is. And connecting it to the bigger picture, this isn't just about fixing things that are broken, though that's huge. It's potentially about enhancing human abilities, understanding ourselves, our thoughts, consciousness in ways that were purely speculative not long ago.
And that really leaves us with a profound thought, doesn't it? As these technologies let us merge mind and machine more closely than ever before, how does our very definition of human start to change? What new kinds of perception or communication or even identity might we discover or create in this nanoscale revolution? Lots to think about. Keep exploring, keep questioning.